Good afternoon. I was asked to provide a perspective of how medical barcoding might be used in an applied way to improve management and restoration efforts on national park and other open space lands. Point Reyes National Seashore was established in 1962, in part to preserve a portion of the diminishing seashore of the United States that remains undeveloped. However, by the late 1990s, the park realized that much of the seashore had already been diminished. Approximately 60% of the park's 2,200 acres of coastal bluff, dune, and scrub habitat had been invaded by two non-natives. These non-natives were European beachgrass and ice plant. We don't know exactly when these species arrived in Point Reyes. However, it was believed that they were planted in the early to mid-1900s to protect adjacent ranches, roads, and homes. Once planted, both of these species have very fast rates of lateral expansion or vegetative growth, ranging from 3 feet in 1 year to as much as 6 to 7 feet in 6 months. Invasives can have both direct and indirect impacts on native dunes. Direct impacts involve invasives encroaching and slowly taking over habitat for native dune plant species, including federally and CMPS listed rare plant species such as Tidestrom's lupin and beachlea. In terms of indirect impacts, invasives displace nectar sources for the federally endangered myrtle silver spot butterfly. Beachgrass can also provide cover for predators of western snowy plover nest, as well as providing cover for mice predators of Tidestrom's lupin seed. Because the park was concerned about these impacts to native habitats and their species, it embarked in 2001 on an ambitious coastal dune restoration program. By 2022, 271 acres of invasives had been removed from 482 acres of dunes. Most of these projects were focused primarily on removal of European beachgrass. We experimented with several restoration approaches. Initially, we tried manual removal, but this was often ineffective due to the fact that the rhizomes can extend down 3 to 12 feet. We also tried mechanical removal, but that was very expensive and resulted in some extensive indirect impacts on adjacent native habitats and ranchlands due to sand remobilization. We finally turned to chemical control because it was more cost-effective and had fewer indirect effects associated with sand remobilization. However, one issue with chemical or herbicide treatment has been the decomposition of standing dead European beachgrass, and its litter or thatch has been very slow. Biomass or thatch persists 2 to 4 or more years post-treatment. The exception to this has been in foredune areas where sand overwash occurs and buries dead beachgrass. In trying to figure out what might make our restoration efforts more successful, we kept coming back to the fact that we often had these remnant native dune islands that had been surrounded by beachgrass or ice plant for decades without being invaded. We wondered if there was some type of biotic or abiotic factor that was enabling these areas to actually successfully resist invaders. It seemed like this resistance factor could possibly have a microbial basis. In an effort to try and answer this question, we began collaborating in 2017 with Albert Barbarin, who was then at UC Davis and who has since moved to University of Arizona. U of A used metabarcoding to evaluate both microbial species richness and assemblages. Sampling was structured to look at intensity of invasion. We had sparsely invaded native dune mat areas, moderately invaded dunes that had largely ice plant with some beachgrass present, and then heavily invaded areas which were mostly beachgrass. The study also looked at different restoration approaches: mechanical removal, and herbicide treatment. Sampling occurred at 3 study sites along the Great Beach: Bee Ranch North in the south, AT&T North Beach, and Abbott's Lagoon in the north. Differences in species richness among sites, invasion intensity, and restoration approach was only documented for fungi. However, differences in species assemblages was documented for both bacteria and fungi. We found a strong overlap between assemblages of both bacteria and fungi in heavily invaded and herbicide-treated dunes. There were differences between mechanically restored and herbicide-treated dunes in bacterial, but not fungal assemblages. Abundances of particular bacterial functional gills varied with invasion intensity. Cellulolytic bacteria was more abundant in heavily invaded areas, whereas bacteria that was associated with either nitrification or fermentation was actually highest in abundance in the sparsely invaded native dune mat areas. So why are we seeing differences for some of these particular bacterial gills? There may be increased cellulolytic bacteria in heavily invaded beachgrass dunes due to higher plant biomass. Decreased nitrification, or conversion of soil ammonium to nitrates may be due to competition with beachgrass roots for ammonium or lower pH in soils, which can inhibit the nitrification process. The changes in abundance of certain bacterial taxa between native and heavily invaded dunes suggest that beachgrass invasion may be causing changes in the way that carbon is being processed. Abundances of particular fungal functional gills also varied with invasion intensity, with abundances of dung saprotrophs and fungal parasites higher in sparsely invaded native dune mat. Heavily invaded areas also had the highest levels of a particular class of arbuscular mycorrhizae, the archosporomycetes. Changes in soil biota with restoration varied depending on restoration approach. For herbicide-treated areas, nitrifier abundance remained low. However, abundance of archosporomycetes and cellulolytic bacteria did decrease. Our results suggest that some impacts from beachgrass invasion have persisted and some haven't. When invasion effects persist after invaders are removed, they are often termed legacy effects. Once established, invasive plants can interact with their biotic and abiotic environment. These interactions can have lasting or legacy effects on biotic and abiotic components of ecosystems. Legacy effects can persist for years or even decades after invasives are removed. The situation with mechanical removal was a bit different. Mechanical removal instantaneously eliminated legacy effects because it involved flipping or inverting highly invaded soils with less invaded soils from below. While this would suggest that mechanical removal areas might have been released from some of the impacts from invasion, a 2013 study found reduced rare plant germination and biomass in flipped soils compared to native dune ones, possibly due to the presence of a parasitic microbe. Our next big question was whether changes in microbial communities with invasion or with restoration was responsible for delayed decomposition of beachgrass. Delays in decomposition of beachgrass could also be mediated by the physical environment, plant chemistry, and soil chemistry. The physical environment at Point Reyes would appear to be conducive to decomposition, as there is high moisture and physical stress from winds along with moderate temperatures. Plant compounds such as carbon, nitrogen, cellulose, silica, and lignin can affect litter decomposition rate. Beachgrass at Point Reyes had a high CN ratios, approximately 71:1. But otherwise, the range of results for more recalcitrant compounds such as lignin fell at the lower end of those reported for grasses. Microbes need nitrogen in the soils to process plant litter that has a high CN ratio. A separate soil study that was conducted by the park concluded that low nitrogen in dune soils may contribute to deceleration of beachgrass decomposition. Obviously, what we are hoping for in most cases is for legacy effects to either not develop or to dissipate quickly. In the case of cellulitic bacteria, abundance in herbicide-treated areas dropped down to levels that were similar to those in native dunes. However, low abundance of cellulitic bacteria in restored areas could be problematic. That bacteria is needed to break down the cellulose in these extensive necromass stands. Reductions in bacteria and other microbes after restoration would potentially suggest that perhaps herbicide treatment is adversely affecting these taxa. However, published studies showed that herbicide treatment either had very short-term adverse or even positive effects on bacterial communities, with most types of bacteria, including decomposers, rapidly rebounding following herbicide application. To tease apart changes in decomposer microbia with invasion and restoration, we conducted a follow-up study in 2020, again in collaboration with the University of Arizona. Sampling was structured to evaluate invasion status of back dune areas in particular. We looked at sparsely invaded native dune scrub. Dune scrub is actually the target native vegetation community for back dune areas. We also looked at heavily invaded beachgrass areas and formerly heavily invaded beachgrass areas that were restored using herbicide treatment. We further stratified herbicide treatment on age or years since treatment. For the follow-up studies, we focused more on the northern section of the Great Beach, including AT&T and Abbott's Lagoon. The data is still being analyzed, but one of the preliminary findings is that wood saprophytic fungi are somewhat lower in invaded dunes than in the native dune scrub. Wood saprophytic fungi are the primary drivers of the decomposition process and perform initial decomposition of cellulose, lignin, and lignocellulose. Following herbicide treatment, saprophytic fungi communities do not appear to rebound in terms of relative abundance. We also looked again at cellulitic bacteria, but this time exclusively in back dunes. Trends here are a bit more difficult to tease apart. Cellulitic bacteria appear to have slightly higher abundances in invaded back dunes than in native dune scrub. Immediately after restoration using herbicide treatment, abundances drop dramatically. However, with time, it does appear that abundances do start to rebound somewhat in some of the older restored areas. Based on these studies, it appears that changes in the decomposer community are likely at least part of the reason for delayed beachgrass decomposition. So given this, how do we use this information to improve success of our restoration efforts, particularly in back dune areas? Our ultimate objective in conducting these studies is to find some type of adaptive restoration measure that could be implemented to improve restoration success. Some of the measures that have been used before in other studies to improve biotic and abiotic conditions of soils are shown in this diagram. We are obviously interested in seeing if we can jumpstart decomposer and microbial communities in general, so we are focused on microbial conditions, specifically whole soil inoculum additions. To confirm whether invasion-related changes in decomposer communities might be adversely affecting beachgrass decomposition, in fall 2021, we developed an experimental microbial inoculation project with U of A researchers. We focused specifically on herbicide-treated back dunes at Abbott's and AT&T of varying ages or time since treatment. We also used surface soils from native dune scrub as our inoculum source. So what are the take-home messages? Simply removing invasive plant species is not enough. We need to consider and deal with legacy effects left by invasives after they're removed. Legacy effects include both biotic and abiotic factors, and there may be interaction between these factors, for example, microbial communities and soil chemistry. Adaptive restoration measures may be needed to improve restoration success, with measures targeted to particular habitats. In herbicide-treated foredunes, there are less legacy effects, but these areas are more prone to secondary invaders, so long-term retreatment is critical. In herbicide-treated back dunes, measure may focus on decreasing necromass and improving decomposer communities. For example, prescribed burning could be used to eliminate necromass. And as discussed before, wholesale inoculum could be used to, from native dunes could be used to jumpstart healthier decomposer communities. Unless we can find and fix some of the underlying problems with invasives removal, we are potentially doomed like Sisyphus to be rolling the restoration boulder constantly uphill. This all may seem a bit gloom and doom, certainly something we don't need at this particular moment right now, so I want to leave you with this. 10 years ago, the area shown in this photo was a dense stand of European beach grass. Now, many of our restored areas support hundreds of thousands of Tyson's lupin, beach leia, and other rare plant and wildlife species. I would like to briefly acknowledge my collaborators in the studies and the funders who have provided financial support.